So let's start the lecture today. So this was the last thing that we were doing, right, in the previous class, that we were looking at the fundamental unit and discussing this. So we had seen that, that we could get either a situation where you have an activation if mu is equal to 1, or if mu is equal to 0, then you get a repression. And this was based on this fundamental unit that I've shown here on the figure. That means if you have a gene, and this is the traditional way of showing a gene uh, in systems biology, where you have a line and then you have an arrow to show that that is the gene that is getting expressed. You can write the name of the gene in this instead of writing gene 1, and it gives you an output, which is the protein that is being synthesized. So this process of whatever is happening here, that is not really shown, but what is shown here is the regulation. So essentially what it means is that you have a protein, which is the protein that is going to be bound to this promoter site, and that is going to be used to induce the synthesis of this gene. And the way you do that is shown like this. If this one is repressing, then you'll have mu is equal to 0, and you have a depressing take place of this gene that is getting synthesized. So that's all that is there in this. All right. Now we look at another motif, which is a little more complex than the previous one. So what you see is that there are two genes that are getting synthesized one after the other, and this is called a cascade. And of course, we have only shown two genes in cascade, but you can have multiple genes in cascades. So gene 1 is producing a protein, which is the protein that is actually linked to this promoter site. That means this protein would inhibit the synthesis of this gene if it sits on this promoter site. Therefore, once it's like the lac operon, it sits on the promoter site, and then you have to have it induced by some kind of a molecule, and that induction is essentially removing this repression that is caused by this, right? So that's the way you show it. And then this gene is getting synthesized and it produces R2, and R2 goes and represses upstream of this reporter protein at the site P2. And this reporter protein could be a fluorescent protein, which is then measured in terms of the signal that is given out as Z here. Okay. So can you tell me what would be the equation that would govern this first one? Would it be the same as the one I've shown you before? Ratnesh, you're mute, is it? Can't hear you, Ratnesh. Have I given the?
Sir, it shouldn't be the same, right?
For this
So
one?
it will be second order equation, like two first order equation.
No, the first part is the first part, just for module 2. Right, this synthesis is our first part, and the second part of course will be different. So let's look at this. I don't know if I have this. I don't have that here in this. Okay, maybe it's not on this. I get confused with the notation I have. So let me look at the next slide. Maybe in the next slide I've given this. Oh no, it's here. It comes as an enter. Okay, not as a link here. So you see, what he's saying is that this is the molecule that is going to dictate how much of R2 is getting synthesized. So the link is between this, and the same synthesis that you see here is going to be shown here. That means this would be like a sigmoidal step function, which is a signal that is showing you an up regulation in R2, right? And of course, R2 will get removed at a certain rate, and it's going to have a basal rate of AK1. Okay, the reason why so AK1, alpha K1, why do you think we have had two factors like this, alpha K1 like this, written out? Because if you multiply it, it should not matter, no? Whether you have 2 into 3 and whether you 6, it's 6. How should it matter? Right? Think about that. So 1 is linked to the rate of the actual synthesis of this, and another one is linked to another parameter which you independently can control, which will say, which may be a function of this, and you could actually independently compute that and then put it here. That's the reason why they have this little flexibility, so that it's not exactly 6 every time. Like I said, 2 into 3 is 6, but it shows you other mechanisms through which this can be obtained, and so it's better to have two parameters multiplied into each other and give you a little more flexibility when you're actually working out the details of or trying to simulate this equation. Now, now you look at R2, and R2 is going to inhibit the synthesis of Z. So if you're looking at the rate of change of Z, then you notice here that it doesn't have the same structure as the top one. It has got that inhibitory structure. So this would be a step down sigma. So it'll go downwards like that, right? So that's the way this would be. And maybe if I use a pen here. So, oh, that has given me the other. All right. So this would be going down, and that is really going up. Okay. So keep that in mind. And steady state values, of course, very easy. You just set the left hand side to 0 and make whatever you wish to use as subject to the formula. That's not a problem at all. All right. Now, there is cascade, which comes in cascade system that comes in control theory. And the use of this is that you could, if you were to look at the way cascades are treated in control theory, you could import that idea and then look at these gene regulatory networks and then discuss or remodel them in the same fashion. Now, I'll just explain this in a little bit and then go back to that figure and then talk about it. So usually when we look at a cascade, there are usually two processes, one after the other, which are actually involved. So that is as GP1 and GP2 is shown here. And there is something that could be a disturbance from outside. And we'll be talking a little bit about this, that there are disturbances also within the cell and outside the cell. So you have intracellular noise and extracellular noise or intercellular noise also, depending upon the situation. And what you're measuring as a signal, that is the output Y. So this could be a reporter protein which gives you a fluorescent signal. And there is a feedback that could be there depending upon the network that you're dealing with. If not, then this feedback will not be there and this just be a straightforward loop. Okay. So what we see here is a forward loop, right? So you see the equations are one after the other and there's nothing that is going back. So R1 goes to R2. Sorry, yeah, I mean I1 goes to R2 and then R2 goes to Z, right? That's what essentially is telling you. So there's R2 and there's no interdependency that is there, that you don't see another function coming up, another variable coming up here into this function, right? So it's just the previous step that is involved. Okay. So keep that in mind, but this is the more general cascade that we normally talk about. And if you looked at control theory for two reactors, then this is what you would see. That there is one system here that is giving something into the other system and the other system is going out and giving you a control. So normally what we will want to do is control the height in this tank depending upon how much you put into the first tank. So likewise here, the analogy is you want to control how much of this reporter protein you want to synthesize based on how much you're giving as the initiator in this case. It would de-repress this R1 from this promoter site P1. Okay. So how does this work out? So if I were to look at this, and of course, this one is not interacting. If I were to make this interacting, then I would get this term here. If this for this system, I would not get this R1A2 system here. How many of you know this? How many of you have gone through this earlier in your courses?
Yes, sir. Like I've done this in control theory.
Yeah, control theory, right? You remember this, Vishantan?
Yes, sir.
What about the others? Have you done this?
Yes, that we have done.
No, sir.
Who's saying no, sir?
No, sir. Sunil.
Huh? Sunil. Sunil is from our department, right? Our school.
Yes, sir.
So I had given that, a book on control and some chapters from it. Have you taken a look at that? No.
Yes, sir, I will do.
Okay, but it's very simple actually. See, this is a linear system. That's the reason why it's simple. So I'll just explain the first one. You will be able to see the answer clearly, right? What is the first one? Just I isolate this tank. And in this tank, if I look at the rate of change of the height, right? What will depend on? It will depend upon this flow that is coming in. So U1 is the flow. And the volume of this change is going to depend upon this A1. So A1 times the height is giving you the volume, right? So, but there's a resistance which is here. So the flow that is going out depends upon this resistance, right? So what you always have this, that you have the output is equal to the way it works out is that this flow will essentially, this input and the output will actually work together to control this height, right? So likewise, you can see that if I were to just look at the first row, so that rate of change of the first one depends upon H1 into this term and then U1 into this term, right? But if I were to look at this properly, then I'm looking at the rate of change of the height. That means if I multiply the left hand side by A1, let me work it out. So just bear with me, those who already know this. I'll work it out so that this student is a little more clear on this, and then I will move forward. So give me 1 second, and I should be able to get to this point here. I'm going to get rid of this out of here. Pointer, I don't know. Let's get rid of that. So can you see my thing now? If I write this, okay, it's not writing on the screen. Oh, okay. Looks at it this way. So size, okay. Oh, what's happening here? I'm going to end the show so that I can kind of draw this here. Okay, just give me 1 second because just so that I can work this out. All right, so just look at this slide here. What happened to this? Yeah, now it's okay. Always get confused with this thing. Let me just do it with this one only. So, all right. So what I was trying to tell you is that, if I look at this D H1.
Sir, your screen is not shared.
Oh. Oh, yaar. Give me a second, yeah. Just get so annoyed with this when it doesn't work. I'm sharing the full screen for time being. Okay, now you can see, right? So DH1, can you see the screen now?
Yes, sir.
Yes, sir.
Yes.
Just
Yes,
give
sir.
me
Yes,
a
sir.
second because sometimes it just gets so cumbersome to do this. Whereas if you're in the class, I just go to the board and do it. All right. So so this dH1 dt is equal to minus 1 upon R1 A1 H1 plus 1 upon A1 U1. I think that's the way to look at, right? And so if I multiply both sides by A1, then you can see that A1 dH1 dt will equal to, and this will become minus 1 upon R1 H1 plus U1. Now this term in the left hand side, this is really the volume. So it's dV1 dt. So it's the rate of change of volume actually. So that's how, so the volume will change, that the volume depends upon what comes in, U1 minus what goes out, Q1, right? So that's what it means. So, and this part, this term which is here is part of that, right? So that's what is happening here. So when you have two equations, that means you have for two tanks, you'll get two equations. And when you combine it in the matrix form, that's what you see here. So you're seeing this, what you see here is really the matrix form of that, okay? Now, because they are not interacting, I will not see this term. So this thing that I see here, this will not be there actually for this tank system. But I'm showing it here as a general case. Now, when I want to look at this system here, which is this gene cascade, which is here, I could also write this gene cascade into this kind of a form. Now, where will the difficulty come when I try to do this? That's my question. So someone can answer this?
So could you like explain the question again?
No, what I'm saying is it was so easy to write for me to put this in this matrix form here. But here, what I don't have the matrix form. I have this form here. If I want to convert that to this form, then how do I do this? And what kind of difficulty would I face?
So because of that inhibitory term, it will cause some difficulty to convert.
No, not because of that. I'm looking at just the mathematics of it. So you see, you are having, a term in the denominator, you have a term in the numerator, plus you have this term, right? This is non-linear.
Yes.
So therefore, what you have to do is you have to linearize. So this one has to be linear.
So we can use some arithmetic,
Huh?
arithmetic, like some.
So you can
I'm
use
just
Taylor
laughing.
series linearization.
Yes, yeah, just take the left hand side. So all you do is you go and here, partial, oops, it's so difficult to control this partial with respect to I1 of this term, which is this term here, right? You bring that here. And if you do that with respect to this, then you have an I1 minus I10 term which will come, and that will become the linear term. Then that linear term will come here. Instead of H1, you'll have that one. And here you'll have R2. So in the second term, so this will become, the first term will be I1. This will be I1. And that next term that you see there, that will become R2. Okay. So if you linearize it, then those terms will come here, and these terms that you see here will be just the Jacobian, the elements of the Jacobian. Nothing but the partial derivatives of the function. So if I call that f1 at the numerator, I1 at some value, and then f1 and R2, it's not going to be there, so it's going to be 0. Similarly, when I look at the bottom, I would have this term going to 0, but I'll have an R2 there. You get it?
Yes, sir.
Well, I think you've made quite a mess of this. I wish if there was a board here I could draw on. Sometimes, maybe it's easier just to do it on that. Anyway, so this is, you fairly get the idea. I think that's not a problem. Anybody who has a problem with this difficulty in understanding this. So you could try actually because this is just a cascade of two such blocks, it's very easy for you to construct this in Simulink. So you should try to construct this in Simulink. Okay, so this thing could go into Simulink. Okay, and then you could see how this whole thing behaves. So what it will give you is that it will give you an I1 that goes up like this, and an R2 or Z that comes down like that. Okay, I1, R2, both of them will. So I1 will be the signal. So I can choose the signal for I1. You can choose like that or like that for I1. And then for this one, which is here, R2. So R2 would be going up like that, and Z would be coming down like this. That's what you would see when you do the simulation on this for these terms. Okay. All right. Now let me go down further. Okay. Now there is a very nice paper that has been worked out by some of the very good guys here. So I'm going to bring up that. Can you see the paper now? All of you?
Yes, sir. We can
Yes,
see
sir.
the paper.
So this
Yes, sir.
paper is written by some of the best people that in the field. You have Rosenfeld, you have Elovich, you have Allen, you have Swain. Okay. So most of you should know Rosenfeld, Allen, and Elovich if you're in the field. And so if you look at, so what they're trying to do is trying to look at the gene regulation in a single cell. Okay. And to monitor that, it's really very difficult. So they came up with a scheme where they could actually have this thing backed up on the same gene. That means if you put a ATC signal that is anhydrous tetracycline, that will relieve this repression from TETR on this site, and then you can have the synthesis of the yellow fluorescent protein, and this is linked to the green fluorescent protein, so you can see both the input and the output. So the reason for doing that is that in the same cell, under the same condition, you can measure, what is the change that is happening in the protein synthesis. So you know what is the signal you get from the outside, and does that give you a very smooth signal of the protein synthesis inside or not is a question. And what they find is that, well, there is a difference which occurs, and this is because of the intracellular noise that is there. This paper is a very complex paper. I don't want to go into all the details of this paper, but what I wanted to bring to you or notice is this particular slide here, which is here. So this GRF that is talked about here is the gene regulatory function. So this gene regulatory function is the one that I showed you in the slide. Clear? So that is giving you this, data points. These experimental data points are there, sorry. And this fill line that is there is showing you that prediction of the gene regulatory function. And you can see it's bang on that, right? It does fit the data very well, but the experimental data has a variation. So for one kind of, one kind of a protein that they're monitoring, which is the ORF2, you have this much variation, which is for this one. And then for the other one, which is you see here, in the blue, you see another one there. Okay, and essentially each lineage that you looked at, those lineages are actually examined carefully, and what you see here are the data points for each of those variations that they are actually working out for various kinds of situations. So the individual point that you see here is the instantaneous production of the cyan fluorescent protein and the CI linked yellow fluorescent protein. So there's a gene, CI gene, which is there, which essentially also helps in so because it is backed up, so it essentially gives you the flexibility of having both of these fluorescences to be visualized at the same time. And so you can see the cyan fluorescent production here versus this axis and how this variation takes place. Okay, so what they are showing you is essentially that there is intracellular noise that is present. So it's not expected, but that's what happens. So over hundreds of data points that they have, they can see this variation takes place. Okay, you can go back and look at the original paper, and then that will help you to understand what's happening. Okay, all right. Yes. Now I'll share that paper also online. Now look at this circuit that is here, and of course I wanted to show you another. I don't Maybe it'll come up a little later. If it does come up, I'll show you another paper after this. So, so the cascade that you see here, there's another way to represent this, and so you can see the representation can be of this kind also, where you bring in a protein that is actually going to be repressing here. You show there is another link that is there to an external protein, and then something getting synthesized and goes forward, and this can go on and on. So you can. It's much more easier to show it like this when you have multiple or larger number of these units sequenced or cascaded one after the other. Now there is a certain kind of logic gate that works here. So this Wise has published a paper in Natural Computing, and you can see that if you have an off circuit here, which means that you bring it here, and you look at the I2, that's the on. So on and off will give you an off. So that depends on whether it's an AND logic. So this is AND or a OR gate. So if it's an AND gate, then it still gives you a 1, and then OR gate, then gives you a 1, and then from there it gives you the yellow fluorescent protein. So it depends on the gating also. We'll talk about this a little later, because the digital logic that is there will essentially dictate whether or not what signal you put and what is the outcome depends upon how these genes are also regulated in terms of the logic gate. And we'll discuss this in another lecture, in a subsequent lecture. Now this person Wise was the mentor or supervisor for one of my students, Subhayu Basu, when I was in biochemical engineering department. Right. So he was my student, and so before he left, he did a lot of work with me. And so the first synthetic circuit was developed by him and Wise, and there's a PNAS paper on this, which I will show you later. So it was published it was presented in the first non-silicon circuits conference, which was held in MIT, and that was in the year 2002. So this paper comes after that. Okay, so some of you might have, I don't know, you think, maybe your age, were you born? How many of you were born before this or after that?
Sir, I am 2002. Yeah.
Huh?
Sir, my birthday is 2002 from. Yeah. Yeah.
Can you imagine that?
Yes, sir. And also, sir, these circuits are like electrical engineers at our IIT do research on that.
Yeah. There is Shaunak. Huh? Shaunak is there.
Yes, sir. Yes.
So he is the person who's doing this work. His supervisor, I forget his name. He had come and given a talk here in IIT, and what they are trying to do is trying to find an equivalent system or develop an equivalent system like the breadboard is there in electrical engineering circuits. How many of you have worked with breadboards?
Yes, sir. Yeah.
Have you done a breadboard
Yes, sir,
circuit design?
Yes, sir.
So how would you have? So the whole question is, if I wanted to create a check out a circuit in synthetic, in synthetic biology, or synthesize a circuit, and before I want to put it into, and this is for drug delivery, for treatment of diseases. Suppose I want to have a small module that goes into a certain kind of cell and alters the circuitry so that it changes what happens inside. It's very far from there, but how will I test it out? The whole question is, if I want to test it out in vitro, how do I test it out? So the equivalence is not to consider a solid surface to be the breadboard, but a buffer solution to be the breadboard. And using that concept, I think that development taken a long. So I think if you go back or maybe if we talk to Professor Shaunak Sen, then we can actually perhaps get a good idea about what's really happening there. Okay, so that's a new and that's a very good emerging area where people are doing a lot of research. Okay, so anyway, so I'm going forward now, and so you can see that this kind of a circuitry gives you a response which is of this kind. So yeah. So you can see when you have a certain amount of ATC, it gives you a red fluorescence, and then the next circuit that comes up with you. So you can see here the events that are taking place. So those are happening here. So one kind of signal is here and the other kind of signal is there. So what it's telling you is that if, of course, you have to go back and look at this paper where they have used TET01 and ATC, TET01 and LAC as the two genes, and they have a circuit like this. So if you put ATC, you get one kind of signal at a certain level, and if you have the LAC also along with it, then you get another kind of signal. So here the intention is to show you that you could get different signals just as you change the amount of ATC, and the amount of fluorescence that you get, how it varies, along with the fluorescence when you're looking at ATC, how the signal changes. And this is just nothing but a representative figure to show you that this is something that is quite doable in a circuit like this. Now the details of this, of course, you have to go back to the original paper, and that we can't spend so much of time in a class like this. So I'll kind of post it so that you can take a look at it. So so this is a simulation of this, and this is the experimental data on which these simulations are superimposed for certain values of the parameters, and then you find that it does give you a very good match. So which means that the equation that we are talking about, that we are used to describe, that like I showed you earlier, are actually doing what it's supposed to do. It has a very good predictive capacity, and that means that the modeling is fairly accurate even at this level. Okay. All right. Now there are different kinds of purposes for these kinds of cascades that are there. Now what I showed you earlier was just a cascade to show you that something is getting switched off. So just one event is taking place. At the end of it, you are essentially using I1 to act on R1, which will then release from the promoter site P1, so that you get the synthesis of R2. When R2 gets synthesized, it goes and attaches to promoter site P2, and then not this one, I'm talking about the previous slide, and then that gives you Z, right? That's what we were doing, and it will just, so what you would see is R2 going up and Z going down. But here, what we see is that at this point, location here, so I hope you can see the arrow as I move it around. So this location that you see, this location, you see that there is an inhibition coming along with an activation from another source, right? So there are two opposing effects that are felt at the same promoter site. Okay. So there is a dynamic equilibrium between S1 and R2, and depending upon the relative amounts of this, there is a certain kind of behavior that takes place at here, which will dictate how much of Z is going to synthesize. So I'm not going through the details. You can read about the details I mean, by doing a simulation like this, you can actually see what's happening. Now, what really actually happens is that this first part that is giving you a signal here, this R1. This R1 also is linked to this another component which is there, which actually will bind to this. So this R1 gives you, and there is a subsequent breakup of this or a change in this to give you an S1. Either it is based upon this combination that gives you an S1, then this S1 becomes an activator, like we saw in the Arabino's circuitry, right? Then this one also is an activator, which we said earlier that we can have a single molecule or a single entity, which could be a protein with a molecule combined together, that actually gives you a dual function, right? A two position at which it can really react. And in this case, that's what we see. And whereas this synthesis of this protein will actually go and inhibit this. So at this point, if you ever see something like this, immediately you should conclude that this would actually give you what is called a pulse signal. And a pulse is usually a trigger. That means it is there for a short time, it just triggers something and it moves, right? So now we'll talk about these pulses later on also, but here we are talking in terms of just looking at how the OD models for this would be, right? And the OD model for this would be this one, okay? I'm going to move this out. I don't know what you see there, but I'm going to, oops, I wanted to just minimize this one further away. Let's move it out in the corner there. At least in my screen, it shows up here. Now you see, so R2 depends upon this S, and also there is a certain rate of removal of this R2. And Z is dependent upon R2 as well as S1. So R2 is, you can see, is actually going to be repressing, whereas S1 is going to be activating. So there are two of them combined together, and this is the reason why you see a pulse in Z, although there is a small decay which takes place. So if I were to use these parameters, then I could actually get a pulse signal. Maybe I have that here in MATLAB. I'll just see if I can go to, it's not showing me up here. Oh, I wish I had that earlier. Oh. Yeah. The pulse emitter. I don't know, MATLAB coming up or not, I don't know. I hope it comes up. It's coming up, okay. Well, while it comes up, so any questions while that comes up? So how many of you have, so I have, yeah.
So I had a small question. So like, how do people like define the equations for like different models?
So that is actually comes from first principles. So here, for example, I can tell you very easily. If you look at this one, so we know that the Hill equation has a certain structure, right? Here.
Yes, sir.
So let's look at R2. So this is R2 based on S1. We know that S1 is activating. So if S1 is activating, we'll get this kind of a structure, right? K1, S to the N1 divided by S to the N1, right? 1 plus S to the N1. Now,
Right.
N1 has to be defined from the experiment. So you'll have to have an experimental information that tells you whether N will be 2 or 3 or 4, something like that, right? Or some fractional value. Similarly, K1 will also come from there. Similarly, K2D will also come from there. And if you know the initial condition, then this can be integrated at least, right? This equation.
Yes, sir.
So that structure is there. So we know that. Now, if you look at this position here, the second point, so you have to define what are you looking at? I'm looking at the rate of change of Z. Now, forget about whether Z is going to be a fluorescent signal, something, some signal, right? Some variable. So you define Z. So what is the rate of change of Z? I just write left hand side, dz dt. Fine, very easy. And what does it depend upon? It depends upon two variables, S1 and S2. And how does it depend upon this? It depends upon this based on a repression from here, which is R2. And I know this from my Hill function. And I know that it's also going to be activating based on S1. So that is this one. Since this is happening at the same site, so just by the probabilistic logic, it will be that one after the other, it has to be both together, multiplied. Clear?
Fine, sir.
Chantan, you could do that way. So that's one way to look at
Fine,
it. Okay. So the other way to look at it is that you more fundamental of this, if one step before this would have been dz dt is equal to some function of R2 and S1 minus KD times Z. Clear? That is very easy. It has to be a function of
Yes,
R1
sir.
and R2 and S1. Now, what kind of function it is, we don't know. If suppose we don't know, then we'll have to essentially do some experiments to find out what the function of R1 and R2, right? So that is the modeling part of it. So, but we do know ahead of time how this works out, right? So if you looked at if you go and look at the literature for two component dependency, if it was just unimolecular, let's say, it would have been some constant times R2 times S1, right? That's what it would have been. But if it is a function
Yes,
that
sir.
is nonlinear, then instead of R2, there will be a function of R2. Instead of S2, S1, there will be a function of S1. Normally, it will be combined. In this case, it so happens that although it is combined, they are separable into two components. That is, this is a component of R2 and that is a component of R1, right? S1, sorry, okay. Now let's look at the pulse generator. So this is the OD model that I've got here. And I'll just run it. I hope it comes up with a pulse generator because sometimes, last time I've changed something to show in the class and I'll not remember the parameters that I did. I hope this is the right one. It's still asking me to wait. There's a plot of x dot versus x, so that would be this one. It's not showing up. So I don't know what happened to this one. Okay, let me look at this one. It is linked. It is working here. I don't know why it's not showing up. Yeah, there you see. So it's generating a sequence of pulses, right? That's because, the way the model has been written out. Otherwise, if I stop why would there be a sequence of pulses? Because I've got a sequence of inputs, right?
Yes, sir.
Yeah. If I give another variation like this, then I will get a different result. So let's look at that. Maybe if I'll just delete that out and I'll just do this. And I'll redo this thing. I'll run it. So it's not giving me a pulse here, it's giving me this signal here. Okay. Let's see the other one. Okay, so that's the signal that you're getting. Essentially, it's a constant signal. So it should be a step-up change. That's not what it is. So if I had so this is a constant. So if you give a constant, it works like this. But if I should have had a step change here, library. So go to sources here. Create company. Zero one. Let me just keep it like that. 1 unit only. Oops. That's the wrong thing. Okay. What am I doing here? This is also not doing that. I have to check why it's not doing that. But essentially, this gives you the pulse signal. So look at what has happened here. So the values must be not in the zone where it gives you that answer. So anyway, why don't you could all check this up and see. It should give you the pulse generator signal that comes up from here. Okay. All right. I think the parameters may be not given correctly here. All right. So anyway, let me go back here. So there are other motifs which are, cascades are there, links like this. So there is a concentration band detection which happens also. So now this one requires to detect like something like that. It requires to have cascades in two different arms here. So you can see that this one loop is going here and the other here, and both of them that meet here are actually inhibitory. So you have an activation in the first step, you have an inhibition in the second step, and an inhibition also coming from the third step here. Okay, and this one, when you model this, oops, it allows you to detect this kind of a gradient. So what happens is, whenever you have chemical gradients of this nature, where you have a green fluorescence protein, for example, against the particular analyte in this case, which is HSL, from this paper that is this taken from. So you have a French flag model which comes in developmental biology, and this is able to detect that. So what is the meaning of this? It means that this circuitry will respond and give you Z only if this concentration of, this rate at which this is getting inhibited, as well as this rate that's coming here, both of them are in a certain zone of values. Only then will give you that. Else it will not respond. So so I'll come back to this since we are kind of running out of time here. So so there is an example here which is there, but this will require a complete session of trying to explain the entire system and then telling what it is. So let me stop here, and if you have any questions, so we'll discuss a little bit about the term paper. So those of you who have, so let me exit from here. So I have got from the groups of Ratnesh and Vishantan. So what about the others? Have you been able to look at the find the term paper? The main code paper for the term paper?
Yes, sir. Like we started out the paper. We will submit by tomorrow. Sir.
Okay. So that is Naveen's group.
Yes.
So we had three other groups, right? Five groups.
Yeah, sir. Sixth group.
We have the sixth group. What about the others? Chael Singh is there. He's in one of the groups, I think.
So he's from our group only. We both are
He's
from
from your group, okay. So please do this, as soon as you can, because if you don't do it within a certain time, you'll be falling behind. Also, pass it through me, show it to me, so that I can give you some feedback, and then you can go ahead.
Yes, me and Ratnesh is in same group.
Okay, all right.
So,
no, one more thing.
So we selected the paper, so we should send you first that paper we selected, or
Yeah, show it to me once, because sometimes what happened is what I find is I'm just trying to mediate so that you don't run into a problem later on. The reason being is that if you show it to me, then I can tell you, well, at least give you a feedback on it. I'm not going to impose anything on you, but if you have 2-3 papers is better, then I can tell you which one to take from.
Yeah, sir, because I thought we should submit the final write-up.
No, I know that, but yeah, that's all. That's definitely there.
Okay.
But
So then,
if you start on something and then you come up with a presentation and all that, that's not really very engaging with the other students, or then it becomes a problem also, right?
Okay,
and
so
also,
then I will share you in next.
It has to be relevant and also related to what we're doing in the class.
Yeah, sir, I will share you in next 30 minutes, because we already sorted out the papers and all. So maybe I'll share in next 30 minutes with you.
All right.
Thank you, sir.
Okay.
Any so other one question? More thing. So in the instructions, you had mentioned that we should limit like the term paper to like 2 figures and 1 table. So is that a hard limit, or so can we like
it's not a hard limit, but the limit is on the pages, so because what if I don't do that, what happens is people keep on adding pages and they have not gotten to the depth of the work. So sometimes the from the core paper that you're following, 1 or 2 illustrations are sufficient to help you. So if you're not using a table, you can use 2 figures or 3 figures, maybe 4 figures, but it has to be within a page. But if you present it in a way that it becomes 4 big pages, I've seen that happen earlier, and then there is not discussion on those figures, then automatically the term paper becomes, less, what should I say, having less merit, because you have put figures, but the figures are not discussed. It's just you have copied it from the paper and put it there, just to show as if you've actually gone through the paper. But then it is not that. It's more important to be able to pick up the code figures, or maybe 1 or 2 figures or 3 figures. Like I said, no, it should all be on 1 page, or at the most, half a page on 2 pages, so that as you're discussing, the whole point is that you show that you understood the material, and the discussion that you're writing is actually having some kind of scientific background and understanding. That's what makes the term paper. So the purpose of the term paper is that only. When you read something, it's like a journal club. Instead of being in oral, you're writing it down, saying what it is. And it helps you to kind of focus on one topic which is of interest to you, and to understand those things, and so on and so forth. All right.
Yes, sir.
So shall I conclude for today?
Yes, sir. So we will have to write in our own words, right?
Yeah, please write in your own words. It's much better. So see what happens if you sit down, and if you and your friends sit down and discuss. The purpose of that is that if you just sit down, the team just sits down and discusses, and you just write down some points, and one person writes it, and the other person, or 1 or 2 people, just correct it. One person takes the leads and writes it, and then the other person is called, no, this is how we should write it. Then you'll find that it becomes very easy to do this. It just can be done in half a day to 1 day. But then if you're not coordinating, if you're not having that discussion that is important with your friends, then it becomes a problem, because then it's somebody tries to finish it quickly, and then there is copy pasting, not understanding the material. But if you discuss it, then everybody's on the same page. So, and nowadays very easy, just do it online, bring up the paper, tell people to read it. If not, then okay, go back and read it one more time. And then if you write 10 points and you write 5 sentences on the 10 points, you have finished a term paper, right? So, and bring it into a logical fashion. So what you're writing should have a logical flow, not a chronological flow. Like, you discuss this today, and then you discuss something else tomorrow, and that should be the sequence. That's not what it is. After all the discussion, what is the logical way of presenting the work that you've understood? Okay. All right.
Yes,
sir.
Okay.
Yes, sir. Thank you, sir.
Okay. See you guys.
Thank
Bye.
you, sir.
Thank you, sir.
Thank you, sir.
Thank you.